Hi, everyone. This is Dr. Jamesia Hoyle, the coordinator for the Next Generation Global Health Security Network. Thank you for joining us today for our next webinar in the biosecurity. Hoyle, the coordinator for the Next Generation Global Health Security Network. Thank you for joining us today for our next webinar. So we are joined today with Dr. Aparupa Sengupta, the Campus Biosafety Officer at the University of California, Merced, and she'll be speaking with us today on CRISPR and gene editing, the biosecurity and biosafety considerations and best management practices. As a reminder, during the webinar, if you do have any questions, you'll see the chat box to the right of your screen. We ask that you type any questions that you may have in the chat box, and we will get to those questions towards the end of the presentation. So with that, Aparupa, thank you so much for joining us today, and I'll hand it over to you.
Thank you, Jamesia. Thank you for inviting me. It's my complete pleasure and honor to present in this forum. And thank you, everybody who joined us today. Hopefully, we will be answering some of your important questions about CRISPR-Cas9 gene editing and its biosafety, biosecurity challenges and management practices. So, Jamesia, I'm going to change my slide to screen share and share my presentation. Okay, just when I want this to work, it just worked 2 seconds ago. Okay, now. All right, so can everybody see my slides? Jamesia, can you see my slides?
Yes, looks good.
Okay, all right, so then I'll get started. So as Jamesia said, today I'll be talking about CRISPR and gene editing. It has been a very talked about topic globally, and we will talk about some of the biosecurity and biosafety considerations because there is a lot of bioterror, association with this technology, and we'll try to address all of that, and we'll talk about the best management practices associated with it. So before I start and get into the presentation completely on the subject matter, I wanted to start this on a little lighter note. Synthetic biology. So synthetic biology is very commonly, it's a household name these days. Everybody talks about it. There are lots of biological engineering things that comes up, and everybody thinks it's the newest school, right? And so, but the question is, what do synthetic biologists do, or what they want to do, right? So basically, if I put it in one sentence, altering the natural selection, altering the natural processes, natural bioprocesses, right? However, what happens if you look at the cartoon, instead of the gene expected in an expected location, the gene insertion, it happens in unknown sequences, mutation occurs, tinkering. However, the new synthetic gene, as you see the smiley face, is, hey, I work. And so, but in any case, it is a newest school. And I kind of thought of putting this idea into my presentation after speaking to my mom, who's a biologist herself, but she like anybody else in the society who doesn't get into the gene editing as much, was asking me this question about CRISPR. And I was like, hey, I'll start the presentation on a lighter note and, kind of introduce the concept, saying, hey, it's the newest school. I do the coolest thing ever, right? So moving on. Let's address the elephant in the room. Why this is the elephant in the room is because CRISPR-Cas9, like I said, it is kind of becoming a household name. People talk about it, people discuss about it, and the news articles, I tried to put 2 important news articles, and both from very relevant sources. One is from the MIT Technology Review, the other one is from the Bulletin of the Atomic Scientist. If you see these news articles, they make the headlines, and people who do not understand the technology go in fear, go in panic, because they're like, what is happening in the world? I have heard this in my family discussions, people who do not understand the technology, and I've seen with my friends. And so we will try to address the elephant in the room to see if we can get to some extent the bottom of it, right? So if you have to look at the left-hand side of the presentation, you see the first human embryos edited in the US that happened about a year ago in a university in Oregon. Now, the question is, Is it the first time human embryos has been edited? No, it is not. China has been doing this work for a while. US could have done the work because US has the capability and the intelligence to do the work, but our regulations here are very stringent. So we are very heavily regulated at the institutional level, state level, and at the federal level mostly, right? So then how gene editing is becoming a national security. So if you look at the news piece on the right-hand side, in about 2 years ago, in April 2016, the US Intelligence Director of National Intelligence, James Clapper, said that this particular technology, CRISPR-Cas9 driven gene editing technology, can very quickly become a national threat, and it has the potential to become a weapon of mass destruction. So you can understand that calling CRISPR a potential technology for mass destruction can create a lot of fear, panic, because people don't understand. Society do not understand. Non-scientists or even scientists in the field who do not understand the CRISPR technology that well, they only can associate that CRISPR with CRISPR and weapons of mass destruction. But is that what it really is? I mean, I am not denying the fact. So I am here, just for the record, not advocating that we should use CRISPR for everything. Neither am I saying that CRISPR should not be used at all. I'm here to give you the facts because science is based on facts. We can never do science and not base it on facts. So I'm going to give you all the facts that I've collected, all the news articles that I've read, all the scientific reviews that I've collected, and leave it up to us to decide with the proper risk assessment, with the proper risk management, with safe science and responsible science, where do we want to go with this? So now the question is genome editing or gene editing new to the world? Is it new? Like all of a sudden, everybody is talking about it. Is it new? The short answer is no, it is not new. It has been there for a while, so it is not a new technology. The long answer is before CRISPR, talons and zinc fingers, and I'm not trying to get into the nitty-gritty of molecular biology, but for the scope of this talk, I kind of have to give a brief idea of these technology. So please bear with me. I will try to simplify this as much as possible because I stress it again that without understanding the science and the facts of the technology, we cannot do correct risk assessment. And without correct risk assessment, there would be bio threats, there would be biosecurity issues, there would be biosafety challenges, right? So if you look at talons and zinc fingers, just so you can get an idea, talons is derived from a Xanthomonas species bacteria. And if you look at the figure on the left-hand side, you will see that basically what talon does is by modifying the amino acid repeats in the sequence in our DNA, users can customize talon systems to specifically bind to target DNA. So the cut is made where it is recognized here, as you see, by FOC1, which is the part of talon, right? It's an enzyme. And then the cut is made and the target gene is inserted there. And then basically the regular recombination and modification happens and it moves on. Now, zinc fingers is on the similar concept. It utilizes FOC1 nuclease, a DNA cleavage domain, and specific zinc fingers, or ZFNs, as the short for zinc fingers, recognizes different nucleotide triplets and dimerize the FOC1 nuclease, and the activated nuclease introduce a double-stranded break, and for the binding side of the target gene, which prompts recombination and modification. So if you leave the technological jargons on side, the clear understanding is that both these technologies edit the gene by making specific cuts where the target DNA with the proper gene construct goes in there, the target DNA binds in the cuts, then the repair takes place, with the regular process of DNA repair and recombination takes place, and it gets inserted in the genome. Then the question is first about CRISPR, right? So I say first because people are talking more and more about it. And we will talk about in depth about CRISPR because that's today's talk is about, right? So CRISPR is actually also derived from bacteria. And CRISPR is actually a defense mechanism of bacteria where it is basically the full form of CRISPR is clustered regularly interspaced short palindromic repeats. So in short, CRISPR, right? No one can remember that big of a name. So what happens in CRISPR, these are the defense mechanism of bacteria where they basically use this to cleave the phage gene or the phage DNA, right? Phage is basically bacterial viruses, so that they can defend themselves from the bacterial attack. Now, the technology can used in the similar way like talons and zinc fingers, where the RNA-guided endonuclease utilizes a short guide RNA to recognize DNA, bind an endonuclease, induce site cleavage, and then again, the recombination and modification and repair occurs. Now, what makes CRISPR so special is the question, right? Why is CRISPR-Cas9 most talked about if all these gene editing technologies have been used for years now, right? For decades. That's because the ease, precision, and availability of CRISPR technology is rewriting the code of life. It's funny how I kind of put this together, right? It's not like it was the way I wrote it is because to make people understand what the power of CRISPR can do. So this basically article that you see here, which I'm indicating is that the easy availability of CRISPR technology is rewriting. What basically this technology is doing is it has the power to make significant changes in the genome in a precise, accurate, easy manner, and most importantly, inexpensive manner. It's very inexpensive and it's very easily done, right? The constructs can be, anybody who has done molecular biology, they understand that creating the construct and having the gene inserted, like the first figure I showed you, right? Most of the time gene is not inserted where it's supposed to be. It's a huge challenge. We have all been there. We have all done that as researchers, right? So if you have done the research, you understand. So, but then this article particularly, I really highly encourage any of you, if you have not read this article, please do. As I was preparing and doing my homework for this talk, I ran into this article, which was published about a year and a half ago now. It is an incredible article. Michael Spector writes it so well. It is about his interaction and his conversation with Dr. Kevin Esvelt. Many of you who have worked in the field of CRISPR-Cas9 and gene editing technologies, Kevin is a very well-known name. He's an assistant professor. He works at MIT Media Lab. The reason I'm talking so much about him is because he, with his mentor, George Church, they have been pioneers of gene drive. Now, what is gene drive? We'll get in a little bit about it. But just so you know, the reason I'm mentioning is because CRISPR, he talks about how it has the capability of rewriting the code of life or challenge the natural selection or manipulate the natural selection, synthesize the, synthetic natural selection and how he does it. And CRISPR being the most inexpensive technology with the power and ease of access and is what is causing it. But the problem associated with it or the challenge associated with it is that CRISPR-Cas9 is not completely understood by many. The risk of CRISPR-Cas9 is not understood by many. And so if you don't understand the risk with the technology, obviously it will come with challenges because every coin has two sides. Any benefit always hand in hand will come with some challenges and risk. And so that's the talk about. So why are we here today? So you see the figure of CRISPR, right? How CRISPR edits the gene and what exactly happens. Don't worry about this figure so much because as we go on with this presentation, I will explain a little more how CRISPR works. Like I said, we have to understand the science a little bit. I will try to make it as jargon-free as possible, but we will explain the CRISPR technology a little bit as we go. But for this purpose of the slide, we are here today, all of us gathered today, because we want to understand a couple of different things. What is CRISPR-Cas9? Why there is a sudden hype about this technology, right? Why do we care about this biotechnological application? I mean, there are lots of technologies with a lot of applications. Then why do we care so much about this particular one? What are those applications? Why is this technology so important to be understood by biosafety, biosecurity professionals and society worldwide? And you see the word society here is extremely important because when we talk about the bio threats and bioterror threats and biosecurity challenges globally, we will recognize today that why society is such an important part of it, right? And the bioterror threats with CRISPR-Cas9, are they for real? does the society understand the benefit versus threat of this scientific evolution? Because biology has gone through a huge evolution in last couple decades, and it keeps going through evolution. It's like biology is a field which is constantly going through evolution. And as many papers you read, you will find that people are constantly saying that biology is the new cool, or is the new technology of the coming era, right? And so it's going through constant evolution. And most importantly, how can we help? That's why we have gathered here, right? To understand what is our role and what can we do to make the process better. So enhance the benefits and limit the challenges and limit the risk, right? So CRISPR-Cas9, as you see, again, this is like a funny picture to keep it light, like how scientists are working on using a Caesar, but that's really not completely a joke. That's exactly what has happened. I have read, a couple of articles, and one of them, I'm pretty sure it's Dr. Esveld, who says that CRISPR is like a new scalpel of biology, right? We can take it, we can make cuts at the exact point we want. So the applications obviously are widespread. As you see, the gene editing applications are there for agricultural, plant biotechnology, food security, gene therapy. There are so many clinical trials that's going on. And the last one is designer babies. And I know a lot of us among us have ethical issues with it, and as rightfully we should, because there are pluses and minuses of the technology, and we should address both. Then the gene drive, and the gene drive, its own beast, and we'll talk about how CRISPR-Cas9 particularly can be used as a beneficial technology for gene drive, but however, it has its own ethical implications, it has own challenges and biosecurity issues. And finally, the recombinant DNA pathogens, that we study so many pathogens, there are so many emerging pathogens that are there which causes infectious diseases. CRISPR-Cas9 can we recognize those infectious genes and alter them. However, again, we are talking about two important concepts here: dual use research of concern and biosecurity. So we'll talk about that. So the CRISPR-Cas technology in general, though, if you see in society, enhancing the quality of life. The quality of life overall in the entire world has increased. And, private sectors and government funding and a lot of, billionaires are investing in it. Like, Bill and Melinda Gates have invested in it recently in London. And then we obviously have a lot of funding here in the US going on the CRISPR-Cas9. NIH funds these research projects a lot. And recently, I think about a year and a half ago, about 2 years ago, actually, Tata Memorial Fund by Ratan Tata, he has actually established with a partnership with UC San Diego, an institute in India, in Bangalore. It's called Tata Institute of Science and Society, Genetics and Society, and they do a lot of this CRISPR-Cas9 gene drive related research for therapeutics and for agricultural biotechnology, which means they are using it for food security, because India and any of the other lot of other countries in the world are going through food shortage, and this can actually significantly improve our food quality and food security, right? Then there are enhancement of food quality. Gene drive can be targeted gene editing for eradication of malaria. That's another reason Tata Institute invested the money, because India is one of the countries where a lot of people die from malaria, and cure of other infectious diseases. So overall, the concept of CRISPR-Cas9 and this powerful technology can enhance the quality of our lives worldwide. Well then, if the quality of life is enhanced, then why the brows are furrowed? That's the question, right? That's because, and I keep saying this for all my talks and all the presentations I give, and wherever I get invited to talk about the subject, I say science cannot continue in vacuum. Science is such a subject, it can never continue in vacuum, right? It always has, you have to see the both sides of the coin. You have to measure the benefits and the risk, and then, manage it properly. In other words, communication is the key, and we'll talk more about it at the end of the slide. But safe science and responsible science can only happen if we are not conducting that in a vacuum. So meaning what? Meaning these days there's spread of knows hows, right? What does that mean by know hows? It is because with the technology, with the way our internet is, with the way all our resources are available, everybody knows how to do everything, right? How many of you have heard about do it yourselves? DIYs, right? Sorry, at this point, I'm like very used to giving, live presentations, and audience say that, oh yeah, we have heard all about DIYs, but I'm pretty sure a lot of you are nodding at your desk that yes, you have heard about DIYs. So what are DIYs? Well, the thing is, all the information out there is so easily available. Technology is so easily available. You can Google, find a vendor who can send you DNA sequences. And DNA sequences doesn't always mean the real DNA sequence of that pathogen, right? They're oligonucleotides. They're mimicking the exact DNA sequences of the same pathogen, which is actually a synthetic DNA. So obviously regulations related to that are there, but they're limited. Because always keep in mind that the scientific pace, the way science evolves, the regulations cannot keep up with that pace always, right? Regulations always come after. So the new technical possibilities and the new scientific, inventions and discoveries and the synthetic oligonucleotide nucleotide sequence information available, the science democratization. I would not say I'm a big advocate of yes and no or any. I don't usually swing towards either side very strictly. However, I am definitely an advocate of safe science. So with science democratization, the DIYs or do it yourselves, I'm pretty sure you guys have heard that news and seen it. It was on all social media that the guy who injected himself with CRISPR-Cas9 construct because he was trying to achieve something. This is happening everywhere you go. This is one example that got out because science is open. Science is available to everybody with the ease of technology, with the, one phone call away, one internet click away, everybody can order anything. More importantly, they're open accessible publications and open source knowledges. And that's where a lot of things gets limited when it is some technology has a dark potential. We'll talk about that in a second when we go to the dark slide. But the point is, all of this is there in today's world. But what is really not there is the open communication about science, limited training and awareness. And you'll see that I have highlighted limited training and awareness in red and underlined it because that is the challenge that we are facing. And keep in mind, for the entire purpose of the slide, I will be talking about in the US context a lot, just because I work in the US and I follow and know the rules and regulations here, and that's what we follow here. However, I'm engaged in international biosafety, biosecurity a lot with my colleagues who are attending today's webinar with me. And we talk a lot about that. This is not only about the US, right? This is about the entire world. Global biosafety, biosecurity is about the entire world. It cannot be limited to one particular nation. So with all the availability that we have, this makes the technology more vulnerable, right? Of course, the technology is powerful, but it's also vulnerable. And it leads to all the different concerns that we have, starting from bioterrorism concerns, biosecurity concerns, weapons of mass destruction concerns, international biosafety concerns, dual use research of concern, and most importantly, the societal ethical concern. These are all the concerns that comes from that because the technology is powerful, but it's also vulnerable. Now, I don't want to get into, like I said to you before when I started, too much scientific jargon or too much details of the science. However, I do have to say, as a scientist myself, that without understanding the basic science, the risk assessment or the challenge associated with it is impossible to understand. So let's review the basics, molecular biology 101. I promise I'll keep it short and sweet, right? So for any molecular biology, synthetic technology, what are the different things that you need? The basic ones, right? There has to be a vector through which you drive the synthetic, the synthetic gene or the sequence. There has to be a gene target. You're trying to target a particular gene sequence, right? It's always a target. The host system, what is that you're using? And by the way, the vector system, it could be bacterial plasmid or viral vector. The reason why am I saying this will make more sense to you when I go to the risk assessment of it, because that's when these all terms will make sense. So if you have a notepad right next to you, please make a note of this. So vector system, why it is important. Gene target, why is it important? The host system, where are you trying to clone this? What are you trying to change? Is it a bacteria? Is it a human cell line? Is it a mouse cell line? Is it a live mouse? is it, most importantly, you're doing it in a human embryo or germline change? That becomes a whole different ethical issue, right? And whether that host system that you're using, is it endogenous? Is it exotic? Is the vector or the plasmid that you're using, where is it from? Is it from a virus that could be a human virus? Or is it a plasmid, which is a regular E. coli plasmid, which has no pathogen or potential high pathogenicity associated with it? These are all important things. So we have to review it. So now that is being said, let's go and see the CRISPR system a little bit. Again, just to keep it simple and sweet, CRISPR has two things associated with it. You see on the red circle on the left-hand side figure, the guide RNA and Cas9. So what happens is the CRISPR can knock out target genes in various cell types, and this Cas9 can be modified to a larger extent to selectively activate or repress target genes, purify specific regions of DNA, image DNA in live cells. And so what happens, if you see on the right-hand side, the mechanism, how it exactly works, if you remember this figure I showed in the beginning when we were trying to target our talk, like why are we doing today's talk? So you see the CRISPR system. Look in the top of it. There is a guide RNA spacer and a Cas9. Now, this guide RNA and Cas9 can be cloned in the same plasmid, in the same vector, or it can be cloned in different vector, right? And so then the target GNA is basically inserted, and with the guide RNA, it goes and binds with the specific site, and a cut is made. Now, why CRISPR is so specific is because the technology in itself has that precision. It can be precisely used correctly because the precision is within the technology, right? So then after the cut is made, obviously break happens in the double strand, right? And then the repair happens. It could be homologous recombination or non-homologous end joining. There could be different ways of recombination that happens, right? So the repair happens and recombination happens, and that's how it gets inserted in the gene, which is basically then followed through, and you basically see the change that we want to see in the gene editing procedure. So that's how CRISPR works, right? Now that we understood how CRISPR works, then what is the problem? CRISPR is so precise. CRISPR is so, specific. It is inexpensive. The problem is, and this is only one of the article, right? Because I have to keep it for, this presentation's sake. I can go on showing you multiple articles. This is just one example that the effect, the problem is there could be off-target effects. We are all human. I mean, the technology is powerful and precise and inexpensive, but only a human is conducting the research. There could be off-target effect. What do I mean by off-target effect? So remember, again, going back to my very first slide when I said I'm starting it on the lighter note, the off-target effect with CRISPR is less, but there is always a chance of off-target effect. Why? Because, instead of inserting the exact target gene, it might get inserted somewhere else. It could create a new gene sequence, which we didn't want in the first place. It could suppress a tumor suppressor gene, or it could eliminate a tumor suppressor gene, which could lead into more oncogenic problems, right? So there are lots of things that are associated with it. And that's why assessing the correct risk at the lab level, and we're talking about the laboratory level at this point, right? At the research level, at the vivarium, doing the in vivo and in vitro experiments. So we have to recognize the risk first. The address risk meaning, what are the off-targeting effects? And every protocol is different. Every experiment is different. I cannot be writing down all the risk for you right now, and you can be making notes and going tomorrow morning and doing the risk of a CRISPR protocol based on what I taught you today. Yes, of course, you will base that off it, but every protocol is done differently. So there is a risk associated with the agent, and then there is a risk associated with the procedure, the experiment itself. So this can be your guide sheet for your protocol assessment or the risk assessment, but this cannot be the only sheet that you do the risk assessment on. So I'll explain a little more. So you have to see what are the off-targeting effects of this particular experiment. What is the delivery system that is being used? Is it a viral vector? Is it a plasmid? Is it of infectious nature? Is the plasmid coming from a bacteria which is infectious nature? Whether it's a virus, is it a retrovirus? Then it is probably more challenging for the human. So then you have to take steps accordingly, right? If there is a needle stick, if there's an injury. The modification that you're making in the system, the host system, where you're making the modification, are they permanent? Are they heritable? Is that in a germ line or in a somatic cell line? What is it? Is it a human? Is it animal? And then animal, then which kind of animal? Multiple genes being targeted in one construct technology. And CRISPR risk, I taught a course with one of my colleagues 2 years ago, actually, sorry, a year ago, and I'm teaching another one with another colleague this year at APSA conference. And this can be an 8-hour class. We can go on and on about the risk and the specificity of it. So this is why I'm telling you, this is a guide sheet for your starting point, but every experiment is different. Then you have to understand the potential risk to the personal for exposure to material or the resulting modified organism. If there's a needle stick, if they splash something, they cut themselves, what happens? What kind of, and you have to assess this risk before starting the protocol. You can't be having active research going on and then assessing all this risk. This is exactly like any chemical risk. When we do chemical analysis, we do hazard analysis before. It comes with the manufacturer's, warning that this is all you have to do before. You can't do this after. And the most important thing that we often miss is the researcher experience and knowledge. So the person who's conducting the research, how experienced they are, because, experience says a lot, right? And how experienced they are of handling this. We want beneficial technologies. I always say that in my training slides, in my presentation for my university, researchers and scientists, that we want to come in the front page of New York Times and Seattle Times and LA Times and whatnot, and for India, Times of India. We always want to do that. What we don't want is to come for a news for bad news, that something bad has happened. Nobody wants that. We don't want that either. So all these things, basically, it's our administrative controls. Now, the other thing is gene drive. Now, gene drive in itself, it's a process of natural selection, right? It's coming from Mendel's time. Charles Darwin has defined evolution, and, Mendel has shown the inheritance pattern and how it happens. So it's nothing new that we're discovering here. What is new here is that CRISPR-Cas9 makes it easier to make heritable changes, which through gene drive, which is a natural process, can be carried forward from generation to generation. And with each progeny, the heritable changes made by the synthetic technology makes more dominant over each progeny, because, as 50% comes from the father, 50% comes from the mother, and through natural selection, every progeny has multiple more copies of the synthetic heritable change that goes on and finally makes a progeny or a generation where we have the entire generation change to the synthetic technology from where we started, right? So now they have those genes that we inserted in them. So basically, we're engineering the natural selection. Now, there's nothing to be scared about it. It's nothing scary about, well, a little bit scary maybe. But the point is, where I started, is that you understand the technology. You go with the technology, you understand the science behind it, you assess your risk at the lab level, and before you go into the field study and release any of these organisms, you have your appropriate barriers associated with it. You have your molecular barrier and gene drive. If any of you are interested in gene editing technology of use of CRISPR in gene drive, I highly encourage you to go to Kevin Esvelt's, who's an assistant professor in MIT Media Lab, his talks, because I am a ardent follower of his talks. I follow his website, I read his articles, because he is a pioneer on it. And he talks openly, honestly, he communicates science completely openly. He always says that I'm not saying there's no challenge associated with it, but the point is, how do we manage those risks after we assess it to go for it? So basically, the gene editing technology using in gene drive, we can engineer the natural selection. It's cool, right? I think it's pretty cool. I think it's very, very cool. But we cannot just go with the cool factor. We have to understand the risk and manage it appropriately. That's the point. So speaking of gene drive, just to show you, and we don't have to go through the slide sentence by sentence, but just to show you the different barriers. And this is actually taken from Akbari et al. And a lot of Kevin's paper actually talks about it, that how barriers can be created at every level of the experience to keep in check. What do I mean by keep in check? Keep in check basically means the technology doesn't go rogue. So people are not the society. So usually you will see with genetically modified organisms, with gene drives, with CRISPR-Cas9 gene therapies, people get scared. They are panicked. They oppose it. They have ecological concerns. They have environmental concerns. They're like, are you guys trying to play God? You will find multiple articles where people are saying, what the heck are you thinking? Are you trying to play God? So the point is, you get society involved, and Kevin's talks and Kevin's publications are all about that. That we are not trying to play God. We are trying to take people along with us and give them all the risk that is associated with it, so that they understand the technology, they understand what we are trying to doing. This is not something. This is not a corporate strategy of trademarking our and patenting our technology and not sharing. We are sharing it so people understand it, society understand it, and professionals, biosafety professionals, and security professionals understand it. Most importantly, regulators understand it, and the government understand it. Because if everybody do not get on board, such kind of technology cannot move forward safely. And that's when we talk about all the challenges with like a bio threat and stuff like that comes in picture. So in general, when you're doing a risk assessment, these are some of the things that you go through. These are some of the things that I came up with while reading papers, right? Function of the target gene. If it's inserted wrong, not in a correct way, what can happen? Expression in plants. If it's expressed in plants, for food security, because it's a huge, I should say, advocacy going on from the scientific field in countries like Africa, India, Bangladesh, Pakistan, Southeast, mostly the Southeast Asia, or even other parts of the world, China, etc. That there should be more food. We have huge population, less food, so much agricultural land, so the quality of the food is low. The quantity is certainly not enough. It's an unfortunate thing that I have to sit here today and say this, that a lot of the world's population, I don't have the exact figure, so I don't want to give you a random figure, but a lot of the world's population still go hungry most of the nights. How unfortunate is that, when we have a technology which can make that change? However, when I'm talking about risk, do you see that I have added the food security transgene protein expression? What do I mean by that? I mean by that, in the research level, when such kind of changes are being proposed, that the transgene protein is being, certain protein is being enhanced in a particular plant, where it will change the food quantity, it will increase in the mass, then we have to see, is that protein expression is expressing something else in the plant, which is causing a problem when it goes through your, when you eat the food and goes through your physiological changes, is it going to cause cancer? I think most people, most ecologically, environmentally concerned people, they don't want to stop a technology. They are stopping a technology because they don't understand what's going on. They don't understand the risk of it. So if we can communicate it properly, if we can assess the risk correctly, manage it correctly on the research level, then to the field level, and then when it's going to the market, and we communicate that with proper science communication, this can change. This attitude can change, and overall, a lot of worldwide problem can change. Same goes with gene drive, right? It's a heritable change. I already said that. And the changing the ecological balance. However, can you imagine if the mosquitoes, which are the biggest vectors of malaria, if we can change the gene sequence in the mosquitoes and make a complete heritable change, so the malaria can be eradicated from that side of the world, which is India, Pakistan, that side, Bangladesh. Can you imagine what can happen in the world? The world can change. We can eradicate diseases for good, right? So that's the point of discussion. Now, when you're doing it in the lab basis, I recommend, as I told you, from the risk of, discussed in my presentation, based it on vector use, the host organism use, and do the risk assessment correctly as a biosafety or biosecurity professional, right? And then you give them the recommendation and feedback, what kind of containment they should use, what kind of biosafety practice level they should use. There are different biosafety practice level, at least in the US. Most of it similarly followed in Canada. European standard, EU has its own standard. And other countries, I'm not going to tease out every, because I was doing a lot of research on the international biosafety security policy. Other countries may or may not have these levels properly defined or implemented, or vigilantly observing it, right? And so those are the problems. So the risk group of the organism, the experiment that you're doing, that should be recommended correctly based on the biosafety practice level, right? And the state SOPs, right? The correct SOPs, how the safe operating procedure of every equipment use, every experiment use, and relevant documents should be advised to the PI meaning the principal investigator who's doing the research. And then most importantly, the waste management. I would definitely say this for India, because I've been there, I've seen experiments, I've done experiments there. The waste management is not as adequate as should be. I'm pretty sure this is true for many other countries too, right? Because after we are done with the experiment, are we autoclaving it correctly? Are we biologically inactivating it? If you're using blood-borne pathogens, if you're using human blood, if you're doing gene therapy experiments, if you're using CRISPR-Cas9 in a high pathogenic organism, are we biologically inactivating it? Are we collecting all the waste as medical waste? Because I have seen through my own eyes hospitals doing research, and after the research is done, they are either autoclaving or not autoclaving and dumping the waste on the streets. What do you think is going to happen from that? So we are researching on something because we want to solve an issue, but we are not doing the waste management correctly. There is no concept of medical waste management. There is no concept of biomedical research waste management, and that's open thrown in the street. What do you think is going to happen? What kind of impact is going to happen? Are we really resolving any issue? No wonder we have so many antibiotic-resistant microorganisms all over in the world, right? So all gene editing technologies, all CRISPR-Cas technologies are not going to change anything if we are not vigilant. So that's the point. Awareness, talking to the society, doing the risk assessment correctly. These are the keys. And finally, there are training required. There are regular audits required. So you cannot just recommend something and not follow up. That's the worst way of managing any project or any technology, right? Hands-on training as required. Now, dual-use research of concern, the reason I wanted to bring this subject on is because CRISPR-Cas9, now it's called a potential biothreat or potentially can be used for weapons of mass destruction. So dual-use research of concern, as WHO defines, is basically a life science research that is intended for benefit, but might easily be, intended to use for biothreat or other nefarious purposes, right? So how CRISPR-Cas9 falls into it, that's because the applications of it, the highly targeted, highly infectious gene editing pathogens, the disease-resistant pathogen studies, the gene drive targeted population, you can clearly see from the discussion we just had that all these things can be very widely applied and can be used for both for the benefit and for the nefarious use. And that's why CRISPR has been labeled as potential DIRC or biosecurity. Now, international biosecurity challenges, the biggest problem is poorly understood challenges. In a lot of countries, and like I said to you when I began, that I will be speaking from the US and a lot. That's because I do my work here. But the point is, biosafety, biosecurity, it's not a national issue. It's not contained to a certain region. We all have to partner. There's lack of training in international biosafety, lack of expertise, and most importantly, lack of international regulatory compliance. So the way US research is controlled, like I told you, in the institutional review boards, where a lot of us sits in different institutional review boards, and we run a lot of institutional review boards, they are so strictly complied with. We follow up, we make sure all the risks are assessed and managed properly. We go back, we check on their labs, we come back, and the PI and the biosafety officer or the biosafety expert and biosecurity expert, they go back and forth. If it's DIRC, it's labeled as DIRC. It goes back to NIH for review. It's required, if it is required. So we do a lot of compliance, but the problem is in the international biosafety, biosecurity world, is it that compliant? That's the question. And so there could be unanticipated or unintended complication avoidance along with intentional misuse. So there could be state actors who would be using it for nefarious purposes, but also you can understand without proper regulations, proper risk assessment, there could be a lot of unintended complications as well, which we can't avoid. And then what is really needed at this point, what I highly recommend, and I was just having a discussion with two of my colleagues, and we were thinking of going a little vocal more and write paper about it. Global efforts and collaborations, they are a must. There is no negotiations on it. It's otherwise the whole effort is a deal breaker. One country can progress and make all the risk assessment in the world as possible, but if there is no cross-border research regulations, international engagement, thorough understanding of emerging technologies, the science behind it, and the risk associated with it, and the understanding, every experiment is different. You can't have a standardized risk assessment and think that that's it. That's what I will go with. That can serve as a standard to you, but it can never serve as the only risk assessment in the world. Risk changes as the hazard changes, and hazards are altered only when the experiments is slightly different. Experiment A from B from C can be different. I have assessed risk for CRISPR technologies and labeled them as 1 and 2 and 3, and depending on what they are, right? So that's the important understanding that we have to have. Remember, powerful synthetic biotechnological innovations such as CRISPR-Cas9 and pathogens do not need homeland security and border clearances. They are not humans. They are not going through visa checks. They are not going through any homeland security checks. They are not going to apply for green cards. They are not. This is open world for them. So any of these technologies, especially in the world of computer science today, in the world of information technology today, do you think they need any border clearances? So we really have to understand as a global community where the importance is and where the challenges lies and where the benefits lies, right? Again, like I said, bringing science closer to life. This is one of the Kevin Eswell talk. Like I told you, I'm a big fan because he is a scientist who communicates openly and honestly, because communicating is the only way, right? Awareness brings proactiveness in society. Ethical concerns must be addressed. We can't just say we are scientists, so we'll do whatever we want, and this is what the technology is, and you accept it. That's not how acceptance happens. Acceptance happens when you're open about it, when you communicate about it. And that's how we can make the society proactive too. All the biosecurity challenges that we're talking about, the bio threat, the weapons of mass destruction, all those things, people will be more aware. So they will be proactive. They will exactly know what's going on in the surroundings. So the society can be helpful. Like I told you, science cannot continue in vacuum, right? And the most important challenge is many developing countries still do not completely understand the adversities associated with the misuse of powerful synthetic biotechnology. People are competing. Science is a competitive field, and there's nothing wrong in it. I am a competitive person. I understand it. Science is a competitive field. However, just because lab A needs to trump lab B and lab C needs to trump lab A, because Professor Smith wants the tenure before Professor Ronald and Professor Mary is competing, and they're all competing with each other. So we'll keep it all hush-hush. In the process, we are creating issues which could be a global problem. I guess that's where we need to understand. It's a collaborative field. We are not trying to be, we are trying to, science is a collaborative field. We're not trying to hide from each other. We are trying to open communicate, right? And that's how we will solve a lot of issues. Otherwise, this weapons of mass destruction issue, the dual use research concern, this may not happen, may or may not happen in the US because of the regulations, but it will exist in the world. If it exists in the world, like I told you, there is no border that's going to stop it, and we all are vulnerable to it. Remember, good science always comes with ethics, compliance, and honest communication. And finally, I would say the risk management and risk strategies, just to conclude the talk, is do the lab risk management, follow the regulatory compliance, institutional guidelines, review and approval, and that's when the science continues in any institution, at least in the US. Again, I'm speaking for the US right now in here. But again, it is not about one nation. There needs international partnerships, symposiums, webinars, joint forums, discussion. This webinar is one of the examples. This is an effort by NextGen, who is, by the way, doing crazy great work. I mean, I'm a big fan of theirs, the way they are communicating this to the world. This is one of the forums. Enhance overall international governance. You have to understand, if the policy regulators, they're mostly sitting government officials. So if they don't understand, if there's no communication, things are never going to change. If things don't change, we'll be always vulnerable. And open communication, putting a stop to the closed door science. That is old. That is not to be followed anymore. Kevin Eswelt, again, I mean, like I follow his talks, says this thing again and again. If we need to progress as a society, we need to put a stop to the closed door science. We can't practice closed door science anymore. Remember, research advances often outpace policy and regulation. I told that to you before, meaning that policies will come after the technology has already evolved a lot, meaning the risk has already out there. So the policy is not going to outpace the science. Science will always outpace policy. So the only way out is to educate people, openly communicate with the society. That's the only way out. That's the only key. So these are just some of the concluding thoughts. Why this CRISPR-Cas9 is challenged, we discussed this. The gene drive has major untapped potential. Do-it-yourself projects are everywhere. So those are the challenges which are there. And public is unaware of implications. That's another issue. That's why so much fear. That's why so much panic that occurs in society. So the key is to understand the technology and then assess, manage, and effectively communicate the risk. Those are the only things that we can do that is in our power for doing a good science, a powerful science. These are some of my references, and this is my contact information. Thank you all so much for watching this webinar, and I thank you for your time, and I will be open for your questions. My information is all out here for my email and phone number, so please feel free to contact me. If you have any questions, if I can be of any use, it will be completely my pleasure. Thank you, everybody, for your time.
Thank you, Aparupa, for a wonderful presentation. I think it really set the stage for opening up the conversation in many areas. So as a reminder to everyone, if you do have questions or comments, please make sure that you are uploading your questions to the chat box at the right of your screen. Luis, our coordinator for the Biosecurity Working Group, is with us as well, and he will be fielding the questions from the chat box. In the meantime, while we are waiting on questions, just want to make sure that we mute the back line so there's not the feedback. But as we are waiting on questions from the audience, Aparupa, I do have some questions for you just to get the conversation going.
Sure.
You mentioned some of the perspectives, particularly from the international arena, some of the limitations in the regulatory compliance and poor biosecurity planning. And I just wanted to know your perspectives on any additional limitations on the use of emerging technology in the developing world.
Sure, Jamesia, there are, I hear feedback, Jamesia. Is it coming from me?
Maybe if you have the YouTube playing in the background, you'll need to just mute that.
No, I don't have YouTube playing in the background. Okay, now I don't hear the feedback. Okay, all right. Sorry about that. So yes, I do have some additional concerns. The concerns being one of the biggest challenges for any of the developing countries would be the resources. The resources are limited. So a lot of these risk assessment and risk management, if you remember the slide where I was saying that managing the technologies and giving feedback to the PI, a lot of times it comes that we give some engineering controls to the PI to be used, right? So that it's secure. There is limited aerosol generation, and even if the aerosol generation is there, it's in a containment. In other cases, we tell them that they should be using such and such plasmid or such and such, for example, I'll give you an example. If CRISPR is being done with lentiviral vector and they're using second generation of that, we say that do it 4 generations, split the plasmids as much as you can, so the risk is low. We do all of that. That's because they have the funding, they have the resources, and sometimes in resource limited countries or developing countries, they may not have it. That doesn't mean they can't conduct the research, but the problem in today's world is, since we're not open to communication, they never get the chance to get the risk assessed and then given them the option that in a limited resource, because I'm a strong believer of a limited resource doesn't mean you can't do the work. You just have to find a way to do the right work in compliance. But maybe so they may or may not have a very highly equipped instrument, but that doesn't mean we cannot enhance some PPE standard and make it possible in their research. But the problem is the risk is not assessed correctly. So of course there are challenges in a resource limited country, but that is no excuse for doing the science and not in a responsible manner. And that's where the communication part comes in picture.
Excellent, thanks for that. So we talk about resources, and I oftentimes like to talk about human resources
Yes.
because I know that those are lacking in some cases. And then just considering this question from a next generation perspective, as more and more people around the world are being introduced to emerging technologies and CRISPR and gene editing, that they may not have the opportunity within their country or where they live or whatever the case to engage. What is your advice to people in the next generation who may be interested in the field? They're just learning about it. How can they become more involved?
So that's a great question, Jameshi. I'm glad you asked it. So the discussion that you and I were just having a couple minutes before we went live is, I think the forum, that the work that NextGen is doing at part of GHSA, it's tremendous. I think we need to continue because we are the next generation. And then there is a next generation that will come, that is coming very closely after us, right? So we are the people. So I think the training, or one biggest thing, if you follow Sean Kaufman's work carefully, he does a lot of these work in Pakistan, where he trains people. So a lot of us who are trained in doing this kind of technology, we just have to somehow volunteer our time and engage as much as possible, partner as much as possible through webinars, through forums. So it may not be physically, may or may not be possible all the time to travel and to go to all these countries with limited resource. If you get funding for that, that's great, but funding is limited everywhere. But if we can engage with those professionals, because I have met some of those professionals, I've met people from Africa, from Pakistan, brilliant people. They're brilliant people. They just need a little guidance. They need the training that we have had received in the US here. We have been fortunate to get the that kind of training. So we just need to connect with them in the forum, this kind of forum like YouTube Live and Facebook Live, and there are so many internet tools, and just communicate with them. And any time we should open our lines. And I personally I'm saying this on this forum, that my lines of communication are open. I'm on LinkedIn. You have my email, phone numbers. I will be open for any communication. If you need my help, if I can help you any terms, any means, with any of your protocols, any of your processes, that I'm here for that. And I always think about it because I also come from India, where the research potential is tremendous, but also at the same time, the regulatory compliance, the GMO that our prime minister is so close to his heart, but there are obstacles, and the obstacles are coming from the not being aware and the risk not being addressed correctly, and society is fearful. And so I think NextGen is already doing a great work. I think you guys should communicate, start, continue doing the work that you're doing, and connect the US professionals to the rest of the world, because we have been fortunate to get the training and we can just pass it on. And those are brilliant scientists, brilliant professionals all across the world, and we can just connect as a community. Like I said, it is not one country. This is a community. We have to be serving as an international biosafety, biosecurity society.
Thank you both for your strong support of NextGen, and also in just the realization that education and open communication is very important. As we are starting this biosecurity working group, Luis and I are constantly having conversations on how to increase the involvement in our colleagues all around the world. So that's one thing that we're very cognizant of. And I think with you on board and multiple other biosafety, biosecurity professionals, that will continue to strengthen both the communication, the open communication around the world, and also just those lines of open education. Some of the things that we oftentimes take for granted in being in some areas.
Absolutely. Absolutely. You have my 100% support.
Yeah. Okay, so we do have a question from the YouTube Live. And so the question is, how do we partner and keep up with countries who have looser regulations and are progressing further in these technologies, so that we can accurately track and know risk?
That is, whoever asked that question, that is a fantastic question. Let me start by saying that, because it is very close to my heart. I ask this question every day because this is a huge problem in countries where the regulations are loose. And that's why I said that international biosafety is more challenged by this biosecurity. So I have a couple of ideas. And to be completely honest, I'm still exploring what's the best way, because I am very involved from my heart. This is biosafety, biosecurity is not a day job for me. I take it it is very close to my heart, right? And so I would say the only way, the starting point, because you can't change governmental policy overnight. I will not name the countries, but just because, but I have, when I was doing my international biosafety research, for this talk and the talk that I'm going to give for APSA International, I found out that regulations are so loose that institutional level, nothing is happening. So how do you change it? I think the only way to change it is to publish, at this point, for those countries, to publish news articles to talk about that, yes, US intelligence has labeled CRISPR-Cas9 gene editing technologies potential, threat as weapons of mass destruction. However, this is how we can do risk. Are we doing this risk? Are we actually at the institutional level? Are we doing this? And what are we doing at the government level? I mean, the discussions can be only brought into table by two things, right? Well, of course, from changing the policy, but change of policy doesn't happen overnight. It can only change, pen. My mom always says this, that pen is the most powerful thing in the world. You can change with that. And so all we have to do is we have to vocalize this. We have to write articles. And articles are been written. A lot of articles have been written in the US. Very important articles. I mean, I wanted to present 15 other articles here. There was no time. But my point is, we have to communicate through articles to general public, to general society. And then the scientists who are who have conscience and who knows what kind of research they do, hopefully from the institutional level, one day it will reach the government level. And that's why I'm saying that international engagement is very, very necessary. And you do it through webinars, you do it through, but if you write it in a news article, like there are always science blogs in there. If you write it in the news article, people will read it and it will start making an impact. The impact doesn't happen overnight, but there has to be a starting point. And that's what I want to do. And I plan to do. Definitely write some of the articles in India and some of the other articles that you and I were just discussing before, to just put it out there in the public to say that.
Aparupa, very interesting presentation. Congratulations. It is always a pleasure hearing you. I have one question. It may be obvious, but there's no obvious question. What measures have been proposed for controlling the access of these technologies, and if it would be necessary to develop specific guidelines for regulate these aspects?
Absolutely, Luis. You cannot. So that's the interesting thing, right? You cannot limit this technology. It's hard. This technology to be limited, to be not available for scientific research. It is difficult, and to be honest, if you ask my opinion, it's wrong. We should not be limiting technology information. What we can do is, and I think we have to start it in the institutional level, and I'm speaking for every country where there is a lab which is doing any biohazard work, gene editing or not. This should start being implying this guidelines. So the risk assessment, couple of slides that I showed you, I have guidelines written. So I'm happy to share those documents with you. The one that I wrote for UC Merced recently, I'm happy to share it because those are open source. I have no problem sharing with everybody. So I'm happy to share with it, but you can use that as a guideline. Those risk assessment, strategies are guidelines, but how you review each protocol will depend on that guideline. So that's not the only thing. For example, a CRISPR-Cas9 using with a bacterial plasmid for a mouse cell line could be very well be BSL-1, and you can do the work on bench, no problem. Just because it's CRISPR-Cas9, it doesn't mean it's a massive weapon of mass destruction threat. It's not like that. It depends what the experiment is trying to do. So my point is, develop a guideline at the institutional level. Safety and responsible science is an important part. Also, I would like to say an anecdote here. Kind of my personal suggestion. Remember, the world is growing and the science and technology is growing with it, and bioscience is evolving very fast. If the world knows that you take your science very seriously, but not so much safety, you are not doing it safely or securely, you will never get that respect in the global level. So I think that will get scientists' interest, because scientists want to be respected. They want their work to be approved by their peers in the other parts of the world. So some countries who do not take their regulation seriously and implement it seriously in the institutional level or the federal level, they will never be taken seriously in the platform of the world, because world is watching. Everybody is watching. How seriously are you conducting your work? How safely you're conducting your work? So I would say start implementing it in the institutional level. Every institution, if every institution implements it, government will be forced to make the policy change. At one point, they will be forced to make policy change. Does that answer your question?
Absolutely, yes. Thank you so much.
Awesome. Well, Aparupa, we are at the top of the hour, so I want to thank you for the presentation. It's been wonderful. Definitely a lot of thought-provoking information. So I'm sure that we will continue the conversation. And thank you for your continued support of NextGen and also for being a pillar of the biosecurity working group.
Thank you. Thank you so much for inviting me. These are things that are very close to my heart. And so, like I said, it's not a day job. I take it very intensely, seriously. And so I'm always happy to be a part of it. And to all our audience who's watching today, please, my doors of communication is always open. Email me anytime, call me anytime, any questions, any resources that I can give you, any training material that I can give you, any guidelines, guide sheets that I've developed, I can give you. I will freely give that to you. It's nothing, there's nothing, rocket science theory behind it. It's just, the training that I've received, I want to freely, distribute it. So there is nothing, no hidden, corporate patent behind it. So please feel free to contact me. I'll be happy to be a part of this effort.
Thank you, everyone, for joining. If you are interested in learning more about NextGen or the Biosecurity Working Group, I encourage you to visit our website. Thanks so much.
Thank you, guys.